Okay, good evening. So, welcome in the new year. Few people are back. So, what do we do today? We somehow haven't got very far last time, but not too many people were there anyway. So, what we talk about today is again RNA-seq analysis, and the other half of it, mainly once you've done all the pre-processing and aligning, what do you do next? So to remind you where we stopped last time, we used this airways data. So this was a data set where they had 4 cell lines derived from airway epithelial cells, and they wanted to see how these cells react to 2 drugs which are commonly used in asthma treatment, namely albuterol and dexamethasone. And we downloaded all the FastQ files, we aligned it using the separate alignment. Then we used the feature count tool in order to count for each gene how many reads map to it in a given gene, and when we quickly run it through DE-seq. And let's have a look again at this feature count table where we stopped last time. Let's see, I think I put it in here. This was the file which we produced with this feature count thing. You might remember from our command line things last time that one should use less in order to look at big files, because it always works even if the files don't fit in memory. If you use less minus capital S, it also doesn't break the long lines but put them like this, so we can more easily see what we have here. We have 1 line for each gene, always identified with its gene ID. Then comes information about the genes in the transcripts like start, end, strand, length, and so on. And then come our various BAM files, so the different alignment files that we had. And let's start by loading this file into R and then see what we can do with it. What you should notice, if we want to load a file into R, if we can open it in a text viewer like less, we can usually load it into R, but we should quickly see how the format is. You see it's a table. The columns are sorted by tab characters, so there's always 1 tab character between each column to the next. And we have here this very first line, which is just a comment to remind us what we've done. We've used feature count to create it with these arguments. And this comment, to show that it's not part of a table, it's marked with a hash sign. So that's a pretty standard thing that you often see, and it can be quite useful. So now I've prepared a bit of a text, and I will now go through it. Let's see if I manage here. And we again use the tidyverse, which you might be used to by now. And the tidyverse has this read function, read CSV, read TSV, and so on. TSV means tab separated values, so a table where the columns are separated with a tab character, as we've just seen it here. Now, where did I put my... Oh, I've closed my... Here it is. And if I now read this file, you see I have to tell it that the comment line which starts with a hash sign should be ignored. We get here this table with 58,000 rows and a couple of columns. I have to click on this arrow here to see the remaining columns. There's start, there's end, there's the strand. For each gene there's several of them because the gene might have several transcripts. But we are no longer interested in where these genes sit on the chromosome, so we simply kick out these columns here, and all we are interested is actually these ones here, these counts. For each of our sample files, you remember maybe these are the sample IDs, and we get a list of reads. Here, for example, in this sample here there were 34 reads which map to the second gene. So if I now want to work with this, the first thing I do is I have here already prepared this line here, which changes the whole thing into a count matrix. Now, this is the first lesson in the year. You might have forgotten a bit of an R, so let's go through this here, what we have in turns. You remember I always mark a bit up to the arrow, but not including the arrow, and try it out. So this thing reads the TSV files as we've just seen it. Now, this we want to, in the end, we want to get to a matrix. And that's, I'm not sure if we've used matrices before. We've so far always used data frames or tables, which are arranged, two-dimensional arrangements with columns, and each column can be different. There can be string columns and number columns and so on. A matrix is a homogeneous thing. It is 2D and it has only one data type, for example, only numbers. And there's a couple of cases where a matrix is more suitable than a data frame, and this is one of these. In a matrix you have row names and column names, which labels the rows and columns. So I first have to say what the row names are, and the row names should be these ensemble gene IDs. Then I have to say that I don't want to have all the columns, but only those columns which start with the word alignment, so they are one of these count files. And then I give it to the S matrix form to change it into a matrix. And then I get my count matrix, and as you see, it looks like this. What I don't like is that here I have these complicated column names, alignment slash, and then the name of a sample, and then dot bam. So just simply release this. I guess even if you haven't seen the string remove function yet, you will probably guess what it does. We take here the column names of the count matrix, so all these names of my 16 columns. From these column names, I remove the string alignment slash and the string dot bam, and I write this back into the column names. And now my count matrix looks like this. This will be the thing we are working with. For each sample, we know now how many reads map onto each of these genes. Our remaining problem is that we can't quite understand these IDs. We probably can't remember what sample number 508 and 509 was. What we do remember, or at least I remembered when I looked at my notes from last lecture, is that we had a run table. And this run table we downloaded together with this raw data from the paper, and it told us what sample is what. I quickly go here on read TSV and read this table, and you see what this table contains. It contains lots of strange columns from this European nucleotide archive. How many megabases the read had, what the experiment ID was, and a couple of other things. What we are probably interested in is this one here. Which drug was the sample treated with, and hopefully somewhere which cell line it was. Cell type, source name, tissue. Okay, there's lots of columns. I've already prepared this here. I shouldn't try to read dalim, I read TSV to be consistent with what I've done up here. So I read all the sample. I select only the columns I actually need, and I've looked through it before. These are the columns we need. The run ID, that's with strange IDs that we've seen in the column names of our count table. The cell line, you remember we used airway epithelial cells from 4 donors to construct 4 cell lines, and we wanted to check whether all these cell lines react the same to these drugs here, and the drugs are either control, dexamethasone, albuterol, or a combination of both. So this is now my shortened run table. What I also do, because it might be useful, is to add 2 new columns. For example, here this column, albuterol, which either is false or true depending on whether there's albuterol in the sample or not. How do I do this? Well, I use the string detect function, which simply detects whether the word albuterol appears in treatment. And I do the same with dexamethasone. Now I have 2 ways how my treatment is encoded. Once here by this treatment variable, and once here by this false and truth, which tells me about each of the 2 drugs that we have. A crucial thing when we do this, because I had these 2 samples from different things, I have my count table, that one here, and I have my run table, and afterwards I will always use both together. And this, of course, will end in disaster if the runs are not in the same order here, in the columns of this count table, as they are in the rows of this run table. So I quickly check this. Are all the run table, the run column of run table, so this here, and the column names of the counts matrix, are these the same? If I check whether they are the same, I get this here. It says true. A short form to check whether they are all true is to say, are they all true? And now we stop. If not, I think we haven't had it yet. You see, nothing happens. That's because they are all true. If they weren't true, it would, as you can guess, stop, namely write an error. That's a very useful thing. Occasionally, if you make assumptions for the following, and the correctness of the following code relies or needs this assumption to be true, to put something like this into the code somewhere, then usually it should just run through and you don't see anything of this. But if your assumption against your own expectation turns out to be wrong, it will stop with an error and say, stop, your assumption that all table 1 is equal to count matrix is not true. And it will not run through, and you get alerted by this. So this is something, a part of what's called in general defensive programming. Defensive programming means whenever you are confronted with a situation where you say, well, it would be really important that this is the case, but of course it is the case because I've done it that way. When you don't say, of course it is the case, but you leave an assumption or a stop if not statement like this to check this assumption. And maybe you later make some changes upstream which invalidate your assumption, but you've long forgotten that you made this assumption, and then you get reminded by that. These kinds of defensive programming things where you always double check whether your samples are still in the right order and so on are very important, and neglecting this is what causes lots of mistakes and errors. Usually you only notice that you made such a mistake after your paper has been published, and when you have a difficult choice whether you want to tell anybody or not. So let's better not get into this situation. Now, today I will present a little bit of my own work. So we'll start with looking at DE-seq2. So DE-seq for differential expression crawling for sequencing is this tool that I developed a couple of years ago, and that sort of is the thing which allowed me to get a foothold in this whole field and gets halfway known. And the successor part, DE-seq2, is something that I started and that when Mike Love, who was a PhD student at that time, finished. And it's a bioconductor package. So bioconductor is a collection of R packages which are specifically made for which are specifically useful for molecular biology. Now, usually all these R packages that you always keep loading with library, usually they come from this thing here, from the Comprehensive R Archive Networks, which is a web page which has thousands of packages. Whenever you need a package which is not installed on your computer, you simply write install.package, and R will automatically download it from this web page. Unless it's a bioconductor package, then you have to get it from the bioconductor web page. There is some you might wonder, why are there now 2 repositories for R packages and only one? If you ask one of the older folks of the bioconductor projects, they will tell you some story involving 2 people who got into an argument, and you can imagine. I actually try to remember, I can't even remember who the 2 people were who argued, but yes, it ended up with this, and they're not disagreeing about how such a repository should be made, and now we have this thing here, and this is the bioconductor page. A few words about bioconductor. So first, it has this long list of software. Install, get support, learn, use here, where you get all the many packages with description and so on. Second, very important, you have here the help part, where you have a support site where you can ask questions. So if you have any kind of problem with data, you can ask here, but please read the posting guide first, which tells you a bit how to ask the question such that you are likely to get an answer and unlikely to annoy people. And very important, each package, let's go look for my package for example, has a so-called vignette. In bioconductor, we have a rule that no package is allowed without any documentation, and the documentation usually comes here in form of a web page or an HTML file which should explain how things work. So Mike and I have been quite proud of this of a vignette I wrote for DE-seq, And that he then continued. You see it's quite long. It covers more or less everything you need to know. And this is why I will be somewhat short in explaining the details today, because once you get into the situation that you really want to do your own RNA-seq analysis, you will have already forgotten half of what I tell you right now anyway. So what you are then supposed to do is take this document, take 2 hours time and read it from beginning to end, or at least from beginning till about the middle, and then it starts getting boring and goes in special cases. If I call my library DE-seq2 case, then you see it loads lots of packages and other parts of bioconductor and gives you lots of warnings about whatever. That's normal, because bioconductor is a big ecosystem of packages which depend on each other and use each other. And that has the effect of that if you load one bioconductor package, you get it loads a lot of other stuff. If you try to install DE-seq without having any other bioconductor stuff yet, it will also automatically download about 50 packages, install them all. So this is something you do over coffee. So the way how DE-seq works, and that's something which is not specific to DE-seq, but it's typical for most bioconductor packages, is that all of the data is put into one big variable which has a bit more structure than just being a table. And then if you subset the data, this will appropriately run through them. I'll quickly show you how this looks like. You remember the 2 things that we need today is the count matrix and the run table. And we put them both together in one thing. And this is a core bioconductor idea, that you have your main data table and your annotations for the data table in one place. If you think about it, this count table is a 2-dimensional table. In each column is a sample, and for each sample we have not only a name but a lot of extra information. So there is an annotation table which tells you something about what each column means, and likewise there's an annotation table which tells you something about what each row means. For example, the gene's ID, but also the gene's name, its function, its whatsoever. So this is why I have to specify both together, and I have to specify a third thing here, which is called the design formula. And the design formula uses values from the run table, namely the ones which should be accounted for when we do our differential expression analysis. And this is, and for now I'm only interested in the effect of dexamethasone. We will ignore albuterol for today due to limited time. And the other thing that we want to account for is the cell line. Now, why there's a tilde in front and what a plus means and what I exactly mean by accounting for something is a problem I want to sort of delay and discuss next time, and when we will talk about ANOVA, or analysis of variance, which is a very crucial concept in statistics, which is not bound to RNA-seq, but which is quite general, linear models or ANOVA, and then I will come back to what exactly this formula means, but we will see this already today a bit. So basically today I will show you the surface view, how it all looks, and we'll explain later what's happening under the hood. So I already have my examples now. So if I execute this whole command here of saying make a data set from this counts matrix and this run table using this design, then I get here this thing. And if you remember, if I just write an R variable, it will just give me this variable, content of this variable. For these more complex so-called objects, you get a more top-level information. So here it gives you a description. This is a DE-seq data set. It has 58,000 rows and 16 columns. It has one matrix of data, which is called counts. It has row names. It has no additional data for the rows. It has column names, and it does have additional data for the columns, namely this stuff here, and so on. As I said, for a moment I want to ignore this issue about albuterol. So I will only look at the data which doesn't have albuterol. So you might remember this thing was a true-false thing. You might also remember that the exclamation mark means not. So this here now means the samples which have not been treated with albuterol. And because DDS0 is some kind of matrix with extra information we can subset using the matrix notation. I'm not quite sure whether we had this before, but you remember the square brackets is for subsetting, so for only getting a part of it. And if you and if you have a matrix, when you have a comma within your square brackets, and before the comma you tell the machine which rows you want, namely all of them, so I don't give any condition, and which columns I want, only those without albuterol. Now I get a new thing, this time with only 8 samples. That's the one we will now run with it. Now, usually we've done a lot of preparation. We spent a whole lecture, 1.5 lecture, aligning the stuff and counting the stuff. And now I spent 10 minutes talking about how to read the data into R. And after all these preparation, the actual analysis is just 1 line. And probably only takes 2 minutes. Or maybe even less. Here you see it does a couple of different steps while running through this. It first estimates the size factors. That's the normalization. And again, normalization, we'll discuss later what this actually means. When we estimate the dispersions, you might remember from our discussion of a t-test that the crucial point in all these tests is that you have to compare between group and within group variation. When we ask whether men are taller than women or whether blue-eyed men are taller than brown-eyed men, we always ask how much standard deviation is there within the group of blue-eyed men or within the group of brown-eyed men. And when we ask, is the difference between the means large or small compared to this within group difference? And the same holds in RNA-seq data analysis. For each gene, we have to ask how much does this gene vary within the control group and within the treatment group. And when asked, is the difference between treatment and control large or small compared to this? And the details are very similar to what we've done before in the t-test, plus some extra steps, which again we'll talk next time. One of these extra steps or these differences is that we call the within group variance. We don't call it variance, we call it dispersion. Why we call it differently has something to do with we have now so-called Poisson noise. Again, we'll see about this in a moment. Yeah.
When you're claiming estimating, do you mean calculating or is it some kind of approximation?
No, we mean calculating. We have a bit in where one annoying aspect of statistics is if you in a science you need technical terms. And in some sciences, like biology, when you need to have a new concept and you want a name for it, you come up with a new name, preferably by chaining a lot of Greek words together, and then you get desoxyribonucleic acid, and then you make an abbreviation. In other sciences, namely engineering and also mathematics, we have the annoying habit of using an everyday word and giving it a specific meaning. So this is very bad in statistics because a word like to estimate has a very narrowly defined technical meaning in statistics. Namely, we've already done this earlier. You remember at the beginning when we had all our heights of male Americans, we wanted to know what is the average height of a population, and when we took a sample and took the average of a sample and asked how close is the sample average to the population average. What a statistician would say is the sample average is an estimate of a population average. So an estimate in statistics parlance is the attempt to get to a number for which we don't have a complete information. And the term inference means figuring out how good our estimate is. So this is why we have here this word estimating. And then we fit a model and do a test. Again, testing, as you've already realized, in statistics is a narrowly defined word, which means calculating a p-value and not finding out whether something works. So, now one thing that is a typical thing about this object-oriented approach of putting everything in one big object is that you always have a function which gives back the object and then you write it back. So my DDS variable before was all the data that we got, and now it's the data that and now DDS is still the same data as before, but with some extra information. So if I click on DDS, it still looks the same as before, but actually there's now additional data, mu, age, cooks, and a couple of more things. And here the raw data now suddenly has a lot of extra variables. Before it was empty, now there is things like the variance, the deviance, the Cook's distance, so all the stuff which we just calculated. And from these 34 different columns, we can extract the ones which are actually of interest to us by just asking what's the result. And now we get the result, and it looks like this. It gives us for each gene something called the base mean, which is simply the average expression of this gene, averaged over the 8 samples. The log2 fold change, which is how much the gene is upregulated or downregulated. So a log2 fold change of 1 means 2-fold up, of 2 means 4-fold up, of 3 means 8-fold up, 4 means 16-fold up. That's why it's log2. And you remember fold minus 1 is 2-fold down, minus 4 is 4-fold down, minus 3 is 8-fold down, and so on. You might now wonder, fold change of what? So first, the term fold change is something which always annoys me a bit. If you compare two numbers, like the expression in the treated samples and the expression in the controlled samples, and you divide these two numbers by each other, you get what the mathematician calls a ratio. When the first biostatistician tried to explain this concept to his biologist colleague, this biologist colleague must have been a not very bright one, because apparently he must have asked, ratio? What is ratio? Why do you have to always talk in such strange mathematical terms? What do you mean by the ratio is 4? Well, I mean, it's 4-fold higher. And what do you mean by ratio 7? Well, it's 7-fold higher. So why don't you just say foldness? This is why in biology we have this term fold change, which from a mathematician's way is baby talk for the word ratio. But we are stuck with this now. We even have the abbreviation LFC for logarithm of the fold change, which in any other science outside biology is called a log ratio. And but whenever you have a log fold change, the question you have to ask is, what does up and down mean? Because obviously you typically will mean up, like higher in treatment and control, and down is lower in treatment and control. But remember, we haven't told the computer which is the control sample. Yeah. So an important thing is always to look first here at this thing which tells us what is the fold change. Usually it would say here dexamethasone, true versus false. So what is the numerator of a ratio versus what is the denominator? For some reason it doesn't say true versus false, it just says true, but I checked, it still means this fold change is dexamethasone versus untreated and not the other way around. And when I say versus, I mean always numerator versus denominator. So up means higher in dexamethasone, negative means higher in control. So what next do we have? Well, you remember we talked about standard errors of means and of differences in mean, and here it's the same. Our log fold change also gets a standard error, and this standard error is then used to calculate a statistic, which is the fold change divided by the standard error. We have used the statistic before when we did the t-test, where we also compared this, and the statistic then is calculated into a p-value, which you also hopefully still remember what a p-value is. But there is an extra thing: we need to adjust this p-value for multiple testing. Why is that? Just briefly, and we get into the details again later. Imagine some evil competitor of yours has replaced your bottle of dexamethasone with a bottle of DMSO, just to annoy you. And now you do your RNA-seq experiment and compare all your stuff, and in reality there was no agent. There was no difference between the treatment and the control samples. And now you are a traditional life scientist who has read in his textbook that everything with a p-value below 5% is true and everything above 5% is false, which of course we don't believe, but let's imagine we did that. What would be our conclusion if we test 10,000 genes? Now, if there is no different, remember the p-values gives us the probability of seeing a difference of what we see here, or even stronger, assuming the null hypothesis of no difference. Now, in my hypothetical situation of our competitor sabotaging our reagent and replacing it with DMSO, we are in the null hypothesis. So the probability to get a p-value of 5% or less is 5%. Hence, if I have 10,000 genes, 500 of these 10,000 genes will be differentially will have a p-value below 5%. And if I use the rule that everything below 5% is considered significant, then I will have produced 500 false positives. Now let's think for a moment what would happen if we now notice our sabotage, redo the experiment, and now we notice that that this time we get 2,000 genes with a p-value below 5%. We would obviously conclude, well, as we've seen before, if there is no real effect, we expect to see 500 false positives at this significance rule. Now, using the same significance rule, we have used 2,000. We've got 2,000, so probably 1/4, 500 of these 2,000, are false positives, and 3/4 are true positives. So once we have a significance rule, we can use the simple logic to calculate a false discovery rate. We might take, for example, let's again say we have 10,000 genes. We might decide, okay, let's be more conservative. Let's only accept as true hits those with a p-value below 0.5%. When we expect to get, in our 10,000 genes, we expect to get 50 false positives. If we now get 300 hits, so 300 genes below our chosen threshold, and we expected 50 of them to be false positives, then we can say our false discovery rate should be roughly 50 of 300, that's 1/6. This is a very simple way how, given a p-value threshold, you can calculate what your expected rate of false discoveries would be among these. And this idea was brought up by Benjamini and Hochberg, who have one of the most highly published paper in this thing, the Benjamini and Hochberg paper of 1995, which suggests this by now have something like 50,000 citations. But the way I've just told you the thing, the idea is pretty obvious and terribly simple. So how did they get so much success by this? Well, before they wrote their paper, nobody ever thought about the concept of, given a list of genes, which percent of them might be wrong. Because nobody ever worked with a long list of genes. Before, people usually maybe checked 10 different cases, like from a qPCR panel or whatsoever. And then if you have a qPCR panel of 10 different primers and you want to see how many of them show an effect of them, you don't ask how many of the 5 panels, which percentage of them might be wrong. You just want to get an overall assumption of, an overall rule of saying, okay, if I use, instead of a p-value threshold of 5%, I use only 1%, then I could be pretty sure that my result will be correct and will not have false positives. This was called the family-wise error rate, that you just say, you have your whole bunch of things, you just want to know what's the probability to make a mistake. And Benjamin Hochberg said, well, but if you have thousands of hypotheses, it's impossible to not have a few false positives. If you test thousands of genes, how could you ever do this? You have to accept that you will have some false positives, and you have to be satisfied with just saying how many of them are false positives. The rest of statistics refused to publish a paper and said, who will ever test so many hypotheses? And then a couple of years later, microarrays came along, and suddenly people checked thousands of things at the same time. And people realized that Benjamin Hochberg realized how to deal with this long before, just in time for this to become relevant.
Do I understand this correctly, that if we have a list of 10,000 hits and 500 of them, like at the p-value of 0.05, if I only have 500 hits, then this means all what I got is false positive?
Exactly. So what people do in practice, hence, is they use, what people should do in theory is that you use much more stringent p-value cutoffs and then use the logic I've just seen to calculate your false discovery rate. What Benjamin Hochberg suggested to make this in an automated fashion is to just calculate what they call an adjusted p-value For each p-value in your list, you calculate, if I would make my cutoff exactly here, all genes with a p-value below that one will be considered significant, all above will be non-significant. And if I then use the logic just outlined, what would be the false discovery rate? And then you write this false discovery rate next to it, and we call this the Benjamini-Hochberg adjusted p-value. And customarily in RNA-seq analysis, we ask for the adjusted p-value to be below 10%, so that we assume that we get a list of about 10% of them. So if I now ask how many genes have a adjusted p-value below 10%, I get 1,300 genes. Of these, probably 130 are false discoveries, and the rest might be true. There's lots of caveats to this last statement, whether this is really a correct way to interpret it, and maybe we got to this later, but this is sort of a standard interpretation of the thing, that we say we have 1,300 genes below at 10% false discovery rate, we get 1,300 genes. We can now go back and check what the actual threshold was, but this is very important. If you don't know about the need to use the adjusted rather than the raw p-value, you run into the trouble that you actually look into an experiment. So your question is something which I've seen in practice, that people got their results, looked at the wrong column, at the normal p-value column instead of the adjusted column, thought they found something, but in reality the experiment has failed, and they didn't realize for months until somebody told them that they looked at the wrong number. I hope this wasn't behind your question, that something like this had happened to you. So now we know that we have 1,300 genes, which is a lot, so we should probably look a bit into what we have and how to trim this down. A customary way to get an overview of this is what's called the MA plot. The MA plot is simply done with the word plot MA, and it looks like this. Control Shift. Now, it's a plot here of two columns next to each other, namely on the x-axis we put the base mean column, so just the average expression. On the y-axis we put the log2 fold change column. And when we put every genes in red, which are significant. Now here by default our DE-seq tries to make some clever logic about what to use as a y-axis, and it usually always gets wrong, so I manually specify the y-axis to go from -4 to +4. Remember, this is log2 scale, so this means 16-fold up and this means 16-fold down. So this is our output. You see there's a lot of genes which are differentially expressed with a lot of different fold changes. So to understand, and we will come back to how to actually interpret this plot again in a moment, because there's a catch in it, that it actually lies to you in a way. But before I go into interpreting, quickly to remember what's happening, I just said this plot here, I used a specific function which is special to DE-seq, but you can do this manually using the plotting that I've explained earlier. Yeah. And that is to use simply ggplot as usual and say I want on the x-axis the base mean, on the y-axis I want to have a fold change, and the color should tell us whether it's significant. So far we don't have a significance column yet, so I first take my result object, I mutate it by adding a new column, signif, and this column should be yes if adjusted p-value is below 0.1. You might also have seen that there is a lot of NAs here in the adjusted p-value, but not in the normal p-value column. Again, bear with me, and I'll explain later what this means. It roughly means that DE-seq2 has kicked out a couple of genes a priori, which are not likely to be significant anyway because they are too weakly expressed. Like if you only see 2 reads against 0 reads, and then we gain statistical power by kicking them out beforehand. But I want I want to use the same color for those which have been kicked out and those which have been deemed non-significant. That's why I write it like this. That it's not, if they are NA or if a p-value is greater than 0.1, then it should be black. So if I use this here, then we get here our sample. This is the same plot as we have above here. Only difference is here we have made these little arrows which show you if something is out of a plotting range, and here somehow these things, they don't even appear. They should appear because I added here out of bounds values should be squished in, but obviously it hasn't worked.
The plotting range is bigger now.
Oh yes, right, the plotting range is bigger, so it has extended the plotting range. But some of them have actually infinite fold change, because we might divide by 0, and they are missing. So, another thing that people usually plot is to not plot the fold change against the baseline, but to plot the p-value against the fold change. I do this here. X-axis is the log2 fold change, y-axis is the p-value, and I might minus log 10 p-value, so that its p-value of 10 to the minus 3 appears at 3, and 10 to the minus 4 appears at 4. I also added another thing. This time I specified the color manually to make sure that it's not red and blue, but it's black and red. Why is this important? You see in a moment. I plot the thing, and this is how it looks like. Why is it important that it's black and red? Because you might be wondering, why is this called a volcano plot? Well, because traditionally we use black and red, and then it looks like a volcano. I'm not sure if it looks like the volcano to me, but somebody had a lot of fantasy who invented the term many years ago. I also think, but yes, it's really spitting out fire. So,
what does this then mean, like on the left-hand
yes.
side?
Okay, so what we see on the left, we see the genes which are down-regulated. On the right, we see them up-regulated. Now, one point here, why this plot is useful is, the question is, which are the most interesting genes? Those with a very low p-value, so those up here on the very top, or those with a very high fold change, those to the right. I quickly change here the scale to 100, and then you see there's a couple of genes with really low p-value. And traditionally people tend to jump on these and say these are the most interesting genes. But I will show you later that this is probably not a good idea, at least in my opinion. And wait, we shouldn't lose them. But to get a bit of the volcano plot gives you a feeling of how significance and fold change are related. Here, for example, is a gene with extreme fold change, but that is hardly significant. And we'll have to understand why this is and what this means. The fold change is the volcano plot is usually used to remind people that this is a problem, and B to fill figures in papers. Often it's not quite that pointful. Before I come back to this issue of what the point of the volcano plot is and how to actually interpret these fold changes, there's one important little issue missing here. When you look at this results table, okay, we have here a couple, you see a lot of points, but we're biologists, we want to know which genes these are. I can take a gene ID like this and can put it into Google, and Google will tell me that it's microRNA 6859, because these are ensemble gene IDs, ENSG, but that isn't really fun to look up each gene like that. So how can we automatically translate the ensemble gene IDs, which are in our count file, into gene names? There's a couple of different ways. Some are more elegant, some are a bit less elegant. I want to show you an easy way, which is maybe not how you would always do it, but how it's easy to understand. If you go onto the ensemble webpage, there is here an area called Biomart. Biomart is a way to download all the database information of ensemble in table form. And I do this here. I say, I want to see here, I choose my database, the ensemble genes. I choose my data set. We are looking at human cells. And now I can here select filters and attributes. At the moment I will go to attributes. Afterwards we will download a table, and in attributes we say which columns we want to have in the table, and in filters we say which rows we want to have. So the attributes I want to have here is the gene stable ID. I don't want the transcript ID and the gene name. If I have the gene ID and the gene name, now I can go to results and see what I get with this. And you see I get here a mapping of gene IDs to gene names. And that's all I need in order to map the two. Just in case we need it later, I can add a few more interesting attributes, for example, the gene description, the chromosome, where it starts, where it stops, and whether it's on the plus or minus strand. Now I have this column here, where I have here a description, microRNA, and so on, where it sits, on which chromosome, on which strand, and so on. Here I can again say TSV file and say download it. And I won't download it probably. And I have done this already ahead of time to download this file. And where are we? Here. If you download it, it always saves it by default under a file with a useful name MART export. It was probably bad style of me to not rename the file. I should have renamed it in something like gene ensemble gene to name mapping, and importantly should have made a note that this whole thing is from ensemble release 96. Making such things is important. Why? Once maybe ensemble changes and your results change when you rerun the script, and when you wonder is this the same version as last time. And also once you write the methods part of your paper, you should give proper version numbers and say these are the gene IDs from ensemble 96. So don't forget whenever you download data to write down, to make a note of a database version. So I load my file. Here you see the column I've just shown you: gene ID, gene name, chromosome name, gene description, chart, and so on.
There was a checkbox: use unique results only. What does uniqueness mean?
Sometimes the same row appears twice, and then you can kick it out. Actually, I've forgotten why it does, but it doesn't make a difference in that case. I usually check it.
As a unique identifier.
And where it appears twice is, for example, and that's the reason why I said I don't want. Where was the gene name? If I have, by default, you always find it like this. And why I deselected the transcript ID. If I do this, then you see now I see every gene with multiple transcripts now has multiple rows. No, you can't see it because this happens to be all single transcript genes, but this way. So, by the way, one attribute which I now forgot and which is very useful is this one here, gene type. Whereas gene types are protein coding genes, linked RNAs, tRNAs, mRNAs, and so on. And often for an analyst, the first thing you want to know is which of these genes are actually coding for a protein and which are doing something else. So it's useful to add this. Now you remember I have my results column here. And I want to add to this results column now extra information to all the extra information from my MART file. So that I add the extra columns from this file here to my results table. How do I do this? Well, I can use the left join command. You remember the left join command from before? It's a command which merges two tables by putting next to each other corresponding rows, and it checks which rows are corresponding by making sure that they agree, in this case, on the ensemble ID. There's one problem. The bioconductor people have done one thing that annoys me terribly, and this is an ever ongoing dispute. They've introduced, at some point they decided that they don't like the standard data frame of R, and they introduced a new way of doing of writing tables, which they call the capital D data frame. It looks the same as a data frame, but it is written with a capital D. If I do this, for example, here you see this is a data frame, a bioconductor data frame. It's different from an R data frame. It's different from a tidyverse table, and the things are annoyingly incompatible. So whenever you get something out of a bioconductor package and you want to go on working with it with a tidyverse, the first thing you have to tell it to use is stable to convert it from a bioconductor style data frame into a tidyverse data frame. The tidyverse people are very opinionated that they don't like row names. They think that having a column like this with row names which doesn't have a column name is stupid, and every column should just have a name. So if I use stable, it will usually take the row names and throw them away because Hadley Wickham doesn't like row names. Alternatively, Hadley allows us a compromise to keep the row names, provided we give it a name. So now the row names have been changed into an ordinary column, and this ordinary column has been called ensemble ID. And now I can take my other thing here, you remember my read TSV, and join these tables. So join this table here to that table here, and I want to join it by matching the ensemble ID column that I made here with the gene stable ID column that I got here. You remember in Biomart the ID was called stable ID. And if I do this, then I now get my results column, which has the ensemble ID, the base mean, the fold change, the p-value, and the gene name, as well as the chromosome, the description, the start, and the end. Now, only as we are at it, I also add the significant column again, which tells us what is significant and what not. Significant again at our 10% false discovery rate, which you should always remember is a completely arbitrary choice to make the cutoff at 10%. And if we now do this, now I have my nice results table. And I can take it and I can now sort it, for example, by the fold change. And look here now at the genes with the strongest upregulation. I also have a reasonable p-value. And you see here, the most strongly upregulated gene seems to be a gene called GBX3. Now at least it looks a bit like biology. The next thing I want to show you is, once we have now genes, we should get a feeling of what actually is behind each of these columns. It's always a good idea to do spot checks and going the analysis backwards. You might not quite know what DEC2 does, and you can now pick one of these genes. Let's take maybe the third most strongly expressed ones, and now this one the fifth most strongly expressed, because that's what I copied here. Call it gene, and quickly plot this specific gene. Here is this specific gene plotted what actually happened. And you see here in each of the 4 cell lines we go up, the gene is strongly upregulated if you go from the control samples to the dexamethasone treated samples. How have I done this thing here? As you see here, first I had my run table, and I put this run table into the DDS object, so this one thing for everything object. Remember, this was the DDS object, this DEC dataset, and the call data function gives us the annotation for the columns. And again, we get one of these annoying bioconductor data frames with all the stuff which we put in. So first thing, I turn it into an ordinary data frame. Now I have that. Next thing, I add a counts table. Now I have to explain this quickly to you. There is a function called counts DDS, and it just gives us back the counts matrix that we put in. And there's another one which is called counts, normalized equals true. What this one does is it gives counts, and each count has been scaled by a sample-specific scaling factor, the size factor, which just accounts for, which is just a bit smaller from one for those samples which have been sequenced especially deep, and a bit bigger than one for those samples which have been sequenced a bit more shallow. Now, with this, of course, this way now I get my count table with a normalization, and it's a matrix. You remember a matrix I can subset with square brackets, and before the comma I say which row I want. I want the row which is labeled with this row name, and I want all columns. And with this I get these columns. And these columns were in the same order as the rows in our call data object. So I can just add this column, I can just add this thing here. I can simply add it to my column to my data frame as a final column. So if I put all this together, now I have here my run cell line treatment, whether it's albuterol and dexamethasone treated, and here the normalized counts. And also appears here this normalization factor. And this I give to geom point, and as usual I say x-axis cell line, y-axis normalized counts, column is the treatment, and I get them here. Here's one last new thing, the log1p transformation. You all hopefully know that the logarithm of 0 is minus infinity. If we want to plot expression data on a log scale, this is a problem because if we have 0 counts, it would appear at minus infinity and we can't see it. Customary thing is to simply add 1 to all the counts, so that 0 counts become 1 and the log of 1 is 0, and 1 counts becomes 2 and the log of 2 is 0.2 and so on. Problem is that then your axis labeling will be wrong, because your axis labeling will be, say, 0 where it should say 1. The other out. And here in ggplot they offer this log1 plus, which does exactly this. It adds 1, takes the logarithm, plots the things logarithmically, but now labels the axis correctly, that the 100 is now really at 100 and not at 101. Which is something that you will notice that most papers do wrong. Whenever they show this axis, they show you the data, and if you look closely, you notice actually they have done log 1 plus. So what they labeled as 0 is in reality not 0 counts but 1 counts. It pays to do this correctly, if not just to not confuse your users. Now, quick advertisement break. Another thing that we've done recently, very recently, we released this in the last couple of weeks. If you now look at this is now a typical gene. What you may want to see is, is this a typical gene? Do all the genes look like this or not? You all know the typical example that you always see in papers. This is a typical example, completely randomly picked. It is not the only gene that looks nice in our whole data set. If you want to avoid this suspicion, or just you want to get an honest view on it, what you should do is make such plots not just for one gene, but for a whole couple of genes. So I should copy and paste a lot of these different genes here out of this thing, put it here, and plot this thing. And that's a bit too much work. I'll show you something better. Just give it one more second. Here is our MA plot as before. And if I now click on the gene, it shows me the corresponding gene. Here, for example, if I now look here at this top expressed gene, which is usually no problem, but there's something wrong with my mouse. What's wrong with this mouse? Aha. This is the top expressed gene. You see they're all nearly 0, and when they go up, here is a typical gene, which is something like this. Here's a gene sort of barely significant, just above the 10% significant thing. Here's one which is just below. Here are, for example, cases where it's inconsistent. The gene goes up in some and it goes down in other cases, and so on. So this is a good way to get a feeling of what doesn't done. So how have I done this app? The traditional way to get this app is to hire a Hiwi, and they use a tool called Shiny, and usually then spend a couple of weeks programming this. Now Svetlana Ovchinikova, who's a PhD student of mine, and I are very proud to have made a way to make this much easier. Whenever you have two plots and you say it would now be easy that I can click on this plot and when the other plot updates, if you want to link these plots, then you can now do this right in R. So remember what we had before is we had our MA plot. Where is it? That one here. And the MA plot here has just code like this with X, Y, color. And we have our single gene plot here also with code for X, Y, and color. And now we put these plots together and use a slightly different thing, which is called linked charts. We say open a new page in the web browser with space for 4 plots. We want a scatter plot, so just a point plot. The x-axis should be the column base mean from the results table. The y-axis should be the column fold change. The color should be according to the column significance. The label, maybe you haven't noticed, but you get the gene name here labeled when I hover over them, should be taken from the gene name column. And it should be on a log scale for the x-axis. And the color legend here should be labeled with significant. So this looks more or less the same as ggplot, slightly different syntax, but the same way. The second plot, here the one on the right, you see this here is placed at place A1. So row A, column 1, and this here is placed at row A, column 2, to the right of it. And here we say the x-axis should be a cell line, The y-axis should be the counts for the chosen gene, log 10 1 plus. As before, the color value should be the treatment, and the title of the plot should be the gene name. And for the counts of which gene, of a gene in the variable gene. And how does the plot know where the gene is? Well, whenever I click on a point here, the web browser tells R that this point was clicked, and R then calls this function here. And what this function does, it gets told where the user clicked on point number K, and it simply saves this point in the variable gene and tells link chart, now please read chart number A2, so the right-hand chart. We are, as I said, quite proud of it because we think that this is really a way to make exploring your data much easier. You can, whenever you have this feeling that you want to drill down into a plot, and there's many cases, you have correlation plots, you might have gene response. We're currently coming up with lots of examples where you have a point and you know each point in this overview plot. So I call this the master plot, and each point in this master plot tells a whole story, which is basically summarizes a whole plot like this. And you can always, and whenever you can write easy code to give to make the master plot and make a specific detailed plot, when you can link them together with our link chart framework. So here is web page for the thing, and we are currently looking for beta testers. So, what I still had a couple of things which I wanted to show you, mainly talking about gene set enrichment, but it is 6:05, so I guess we talk next time about gene set enrichment and LFC shrinkage. Just for you to see an overview of what we've done. What we have to do next time is, you might all know this concept of gene set enrichment or GO enrichment, where you have your long list of by now 1300 genes. We want to know what do they have in common, and we will find next time that the thing that they all have in common is that they are known to be related to the epithelial to mesenchymal transition, and that they are related to the extracellular matrix, so that we have especially many secreted proteins. And how do we use the gene ontology database and the molecular signatures database and the signaling hallmark list and all these things? That's what we'll talk about tomorrow. The other thing is, and that I show you very quickly before finishing as a teaser, is I told you earlier that this MA plot is misleading. And if you look at it before, you might have already noticed that it has something which is suspicious, and that is what I call the trumpet. Notice here that this decision boundary has this strange shape, and also maybe even more suspicious is that the fold changes for the weakly expressed genes seem to be much more strongly. There's loads of genes with 5-fold, 10-fold change, which only have 2 or 3 counts, and many of them are not even significant. And that is no wonder at all, because if we are here at the range of an average of 3 or 4 reads per gene, then having 1 read more will completely change the fold change. And having 1 read more in each of them can quite easily bring us accidentally up there. So just the fact that a single read can completely change the fold change means that these fold changes for the weakly expressed genes can be completely exaggerated. And if we are now looking for the most strongly affected genes, so those which are most strongly react to dexamethasone, then we will end up being misled by them, and we'll pick, for example, these genes here, which only are so high up because they are so much weaker in expression, while here we can get the fold change more exactly, and hence we will have less variance. And what we added to DE-seq recently, or what we started adding already then, and Mike, who by now has his own group, has now added, is something just recently published, is something called fold change shrinkage, which tries to shrink the fold changes towards 0 by an amount which is dependent on how variable or how imprecise the fold changes. So the left genes, which are weakly expressed and hence more subject to this exaggeration, they get shrunken more strongly towards 0, and the MA plot then completely changes its shape towards this here. You see, now this has vanished. Now we have about the same strength of fold chains all across the dynamic range. It no longer looks like that. The genes here react much more strongly than here, and there's now genes with high fold chains. They probably really have high fold chains. And this now gives us another way of understanding these 1300 genes. 1300 genes are way too many to manually understand. One way is to use the enrichment analysis. The other way is to notice that most of these 1300 genes here have a change of less than twofold. But there's a rather small subset of maybe 50 genes which go up fivefold or even tenfold. And it might be worthwhile to look at these manually and try to reason about them. But for these you have to pick the right one, and this is where the shrinkage is important. So these are the 2 points which I still had prepared and can't do today. The other points which I have promised and also haven't prepared yet is explain to you how the normalization works and how exactly this test and this shrinkage and so on works. Okay, thanks so far, and see you next week, if I remember right, and the week after we make a break. So we will now roughly get to a modus of maybe we make another 3 or 4 lectures, but I spread them out over 2 months because A, I'm too busy to prepare a lecture every time, and B, the room is booked a couple of weeks. But this is now prepared, so we'll discuss it next week, same time and so on. And yeah, so far, thanks for your attention. See you then. Is everybody on the attendance list?
Are the lectures being recorded?
Yes.